The anti-aging supplement industry generates over $60 billion every year. Walk into any store and you'll see shelves filled with capsules, powders, and extracts, each promising longevity, cellular repair, or age reversal. But if you look closely at the ingredient labels, a different story begins to emerge. Curcumin, resveratrol, EGCG, these compounds don't originate in laboratories, they come from plants. The curcumin inside a capsule began in a turmeric root. The resveratrol in a longevity formula existed first in grape skins. The EGCG in green tea extract was once inside a simple leaf. Supplements isolate these compounds, concentrate them, and sell them back at a markup that often exceeds 500 to 2,000%. What's often overlooked is this. The original source, the plant itself, is accessible, growable, and in many cases more biologically effective in its natural form. This is not an argument against supplementation. It is an argument for understanding origin. Because when you understand where these compounds come from, you begin to see something far more powerful than a product. You see a system. The plants in this video were not selected based on trends or traditional claims. They were selected based on documented, peer-reviewed mechanisms directly tied to cellular aging. Not vague phrases like supports health, but measurable biological processes, telomere dynamics, mitochondrial function, cellular senescence, chronic inflammation. These are the mechanisms that determine how fast you age at the cellular level. And many of the most studied compounds affecting these systems are found in plants that can be grown in a backyard, a container, or even a windowsill. In some cases, the version you grow yourself, harvested fresh, unprocessed, delivers higher bioavailability than its manufactured counterpart, which reframes the entire equation, not as consumption, but as cultivation. Because the question is no longer what to buy, it becomes what to grow. And once you understand the mechanisms behind aging itself, the role of these plants becomes far more precise and far more powerful. The first plant on this list is also one of the most extensively studied compounds in all of nutritional science. Green tea. Derived from Camellia sinensis, it has been consumed for over 4,000 years. But its significance in modern longevity research comes down to a single compound, EGCG, epigallocatechin 3-galate. This molecule has been studied in thousands of peer-reviewed papers, making it one of the most validated plant compounds in molecular biology. Its effects operate across multiple aging mechanisms simultaneously. First, telomere maintenance. A large population study found that individuals who consumed green tea regularly had significantly longer telomeres compared to non-consumers. Each additional daily cup was associated with measurable slowing of cellular aging. This is not theoretical, it is observable at the chromosomal level. Second, activation of SIRT1. EGCG activates a group of proteins known as sirtuins, particularly SIRT1. These proteins regulate some of the most critical longevity pathways in the body: DNA repair, mitochondrial production, inflammation control, cellular stress response. SIRT1 is also activated during caloric restriction, one of the most consistent lifespan extending interventions in biological research. EGCG partially mimics that response, without requiring dietary restriction. Third, antioxidant and anti-inflammatory protection. Unlike basic antioxidants that act once and are depleted, EGCG influences cellular signaling pathways that reduce oxidative stress at the source. It doesn't just neutralize damage, it reduces its production. Now consider the growing advantage. When green tea is commercially processed, stored, and transported, EGCG begins to degrade. Time, heat, and oxidation reduce its potency. But when tea leaves are harvested fresh and prepared immediately, compound levels remain significantly higher. This changes the delivery entirely. Instead of a degraded extract, you are consuming a live compound at peak concentration. Green tea plants can be grown in containers, controlled environments, or suitable outdoor climates. A single mature plant can provide a continuous supply of fresh leaves, which turns a daily habit into a direct, renewable source of one of the most studied longevity compounds available. And this is only the first plant. Now here's where this changes. If one plant stands out for addressing all four aging mechanisms at once, it is turmeric. Its active compound, curcumin, interacts with four key pathways. First, inflammation control by suppressing inflammatory signaling. Second, mitochondrial protection, supporting cellular energy and vitality. Third, alleviating senescent cell impact, creating a quieter cellular environment. And fourth, telomere preservation, slowing the shortening of our DNA caps. To unlock these benefits, bioavailability is key. Combining turmeric with piperine from black pepper increases absorption by up to 2,000%. A simple, ancient formula with a profound scientific impact on every major aging pathway identified so far. Aging is not driven by biology alone. It is also accelerated by stress, and few plants address this connection as directly as ashwagandha. Derived from Withania somnifera, this adaptogenic herb has been used in traditional systems for centuries, but recent research has clarified its specific impact. Its effect on cellular aging is measurable. The central mechanism is cortisol reduction. Cortisol is the body's primary stress hormone. In short bursts, it is useful. but chronically elevated cortisol accelerates telomere shortening and impairs mitochondrial function. It activates inflammatory pathways, amplifying aging. Ashwagandha interrupts this pattern, reducing cortisol levels significantly. This reduction lowers oxidative stress on DNA and slows telomere attrition, as observed in randomized controlled trials. Beyond stress, it offers neuroprotection. Its withanolides protect neurons from oxidative damage and support synaptic function. By reducing pressure, ashwagandha slows multiple aging pathways at once. But the next plant works in a completely different system. Most anti-aging strategies focus on the body, but aging is just as visible in the brain. Memory declines, processing slows, neural connections weaken, and this is where blueberries stand apart. Derived from Vaccinium species, blueberries contain a class of compounds known as anthocyanins. These are the pigments responsible for their deep blue color, but their biological role is far more significant. They cross the blood-brain barrier. This is rare. Most plant compounds never reach brain tissue. Anthocyanins do, and once there, they accumulate in critical regions, especially the hippocampus, the area responsible for memory formation and learning. Their primary mechanism is BDNF activation. BDNF, brain-derived neurotrophic factor, is a protein that supports neuron survival, growth, and connectivity. It is essential for maintaining cognitive function, and it declines steadily with age. Anthocyanins stimulate BDNF production, which means they don't just protect existing neurons, they support the formation of new ones and strengthen the connections between them. This leads to something unusual, not just slowing decline, but measurable improvement. In controlled human trials, older adults with mild cognitive impairment who consumed blueberries daily showed significant improvements in memory performance. Word recall improved, learning capacity improved, even mood markers shifted positively. This is not theoretical protection, it is functional recovery. There is also a secondary layer, oxidative damage. The brain is highly vulnerable to oxidative stress due to its high energy demand. Anthocyanins act as powerful modulators of oxidative pathways, reducing damage at the cellular level, protecting DNA, stabilizing neuronal membranes. Animal studies extend this further. In model organisms, blueberry compounds have been associated with measurable lifespan extension. While not directly transferable to humans, the consistency of these findings supports the underlying mechanisms. From a growing perspective, blueberries are accessible. They can be cultivated across a wide range of climates. They produce annually, and once established, they provide a continuous seasonal supply. Fresh fruit, harvested at peak ripeness, contains the highest concentration of active compounds, which again shifts the advantage toward cultivation over consumption, because in this case, the plant is not just supporting longevity, it is preserving the system that experiences it. And this is where the system expands further. Most compounds labeled as antioxidants work in a simple way. They neutralize free radicals, one molecule reacting with one source of damage. But rosemary operates differently. Derived from Salvia rosmarinus, it contains compounds that activate a far more powerful system. NRF2, the master regulator of cellular defense. Instead of supplying antioxidants directly, rosemary turns on the genes that produce them inside your own cells. This changes the scale entirely. NRF2 controls the expression of over 200 protective genes, including those responsible for producing superoxide dismutase, catalase, glutathione peroxidase. These are not passive molecules, they are catalytic systems, each one capable of neutralizing thousands of reactive species. The key compounds are carnosic acid and carnosol. When these compounds enter the body, they trigger NRF2 activation, which then initiates a cascade of protective responses: reduced oxidative stress, lower inflammation, improved cellular resilience. Instead of reacting to damage, the cell becomes better prepared to prevent it. This is a shift from defense to readiness, and it is one of the most efficient strategies in longevity science. But rosemary introduces something even more accessible: inhalation. When rosemary is cut, crushed, or heated, it releases volatile compounds into the air. One of these, 1,8-cineol, can be absorbed simply through breathing. Studies have detected this compound in the bloodstream after exposure to rosemary aroma alone. And more importantly, higher levels have been linked to improved cognitive performance, which means exposure does not require ingestion. The compound enters through the air, a rare delivery pathway among plant-based interventions. From a growing perspective, rosemary is continuous. It can be harvested daily, used fresh in cooking, or simply present in the environment. Small exposures, repeated daily, activating one of the most powerful defense systems the body has, and the next plant moves deeper into energy production itself. Energy production is one of the most critical determinants of how the body ages. At the center of that system are mitochondria, and ginseng directly targets their regeneration. Derived from Panax ginseng and Panax quinquefolius, this plant contains a class of compounds known as ginsenosides. These compounds interact with one of the most important regulators of cellular energy, PGC1-alpha. PGC1-alpha is often described as the master switch for mitochondrial biogenesis. When activated, it signals the cell to produce new mitochondria. Not repair, not protect, create new ones. This is the same pathway activated by exercise, fasting, and cold exposure. Interventions consistently linked to improved longevity. Ginseng activates this pathway at the molecular level, without requiring those external stressors. The result is measurable. In controlled studies, ginsenosides increased mitochondrial density and improved ATP production efficiency. More energy, less oxidative waste, a more stable cellular environment, but the impact does not stop there. Ginseng also addresses a lesser-known aspect of aging, immunosenescence. As the immune system ages, it becomes less effective. It responds slower, it produces more background inflammation, and it becomes less capable of clearing damaged or senescent cells. This creates a feedback loop that accelerates aging. Ginseng interrupts this decline. Clinical studies have shown improvements in natural killer cell activity, T-cell function, overall immune responsiveness. This is not just immune support, it is restoration of function in an aging system, which connects back to the core mechanisms. A stronger immune system reduces chronic inflammation. It improves clearance of dysfunctional cells, and it stabilizes internal signaling, all of which influence how fast tissues degrade over time. From a cultivation perspective, ginseng is slow. It grows in shaded woodland environments, and the root, the primary source of its active compounds, takes several years to mature. This makes it different from fast-growing plants. It requires patience, but that slow growth reflects its biological role. Deep structural support, not quick stimulation. A long-term investment in cellular capacity. Because when mitochondria function efficiently, every system that depends on energy begins to improve. And that includes almost everything. Now the next plant targets something even more precise. If telomere shortening is one of the core drivers of aging, then the ability to slow or even reverse that shortening becomes highly significant. Astragalus targets this mechanism directly. Derived from Astragalus membranaceus, this plant contains a compound that has drawn intense attention in longevity research, cycloestrogenol. Its function is highly specific. It activates telomerase. Telomerase is the enzyme responsible for rebuilding telomeres, adding nucleotide sequences back to the ends of chromosomes. In most adult cells, telomerase activity is low, which is why telomeres gradually shorten over time. Cycloestrogenol changes that. In controlled studies, it has been shown to activate telomerase in human immune cells. This is one of the few documented cases where a plant-derived compound directly influences this enzyme. And the implications are significant. Maintaining telomere length allows cells to divide more effectively, it preserves function, and it delays entry into senescence. This is not a broad effect. It is targeted, specific, and measurable. Astragalus has already been commercialized in a different form. A compound derived from its root is used in high-cost telomerase-activating supplements, often priced in the range of hundreds to thousands per month, but the origin remains the same, a plant that can be grown, harvested, and used directly. There is also a broader context. Astragalus contains additional compounds, astragalicides, that support immune function and reduce inflammation. This creates a layered effect, direct telomere support, combined with improved cellular environment. From a growing perspective, astragalus is accessible. It grows in full sun, requires minimal maintenance, and develops over multiple years. The root, the primary medicinal component, is typically harvested after several seasons of growth. This delay is part of its function. The compounds concentrate over time, which aligns with its biological role, long-term cellular preservation, not immediate response. Because maintaining telomeres is not about short-term performance, it is about extending the functional lifespan of cells. And that is one of the most direct ways to influence aging itself. But the next plant amplifies protection at an entirely different scale. Some of the most powerful anti-aging compounds are not found in rare plants. They are found in common vegetables at specific stages of growth. Broccoli is one of them, but the real concentration of its active compound appears not in the mature plant, it appears in the sprout. At just 3 to 4 days old, the compound is sulforaphane, and its primary function is the activation of NRF2, the same master defense pathway seen with rosemary, but at significantly higher potency. When sulforaphane is activated, it triggers a broad genetic response. Dozens of protective genes are turned on simultaneously. This includes antioxidant systems, detoxification enzymes, and anti-inflammatory pathways. But the concentration difference is what changes everything. Mature broccoli contains measurable levels of sulforaphane precursors, but broccoli sprouts contain 20 to 50 times more. This means a small handful of sprouts can deliver the same biological effect as a much larger quantity of the mature vegetable. The mechanism depends on a conversion process. Sulforaphane is not stored directly, it is formed when two components meet: glucoraphanin and the enzyme myrosinase. This reaction occurs when the plant is chopped, chewed, or crushed. But there is a limitation. Myrosinase is heat sensitive. Cooking destroys it, which means cooked broccoli produces significantly less sulforaphane. There are precise solutions to this. Eat sprouts raw, or allow chopped broccoli to sit for around 40 minutes before cooking, giving the reaction time to complete. Another method is adding raw mustard seeds to cooked broccoli. Mustard contains active myrosinase. It restores the conversion process. From a growing perspective, this is one of the most efficient systems available. No soil required, no outdoor space needed. Broccoli seeds can be sprouted in a simple jar, harvested in 4 days, and consumed immediately. This creates a continuous cycle. High-potency compound production, on demand, with minimal effort, and the effect is not isolated. Sulforaphane also influences mitochondrial protection and may reduce the impact of senescent cells through NRF2-mediated pathways, which places it across multiple aging mechanisms, all from a seed in less than a week. This is not slow cultivation, it is rapid biological activation, and the next plant targets the quality control system inside the cell itself. Producing energy is only part of the equation. Maintaining the quality of that system is just as important, because over time, mitochondria become damaged, they lose efficiency, they generate more oxidative stress, and if not removed, they begin to disrupt the entire cellular environment. Pomegranate targets this exact problem. Derived from Punica granatum, it contains compounds known as elagotanins. These are not active on their own, they require conversion. Inside the gut, specific bacteria transform elagotanins into a compound called urolithin A, and this compound activates a process known as mitophagy. Mitophagy is the selective removal of damaged mitochondria. It is the cell's quality control system. Instead of allowing dysfunction to accumulate, the cell identifies defective mitochondria, breaks them down, and recycles their components. This clears space for new, efficient mitochondria to take their place. This is a critical distinction. Some compounds increase the number of mitochondria, but urolithin A improves their quality. It removes what no longer works. In human clinical trials, this effect has been observed directly. Older adults who received urolithin A showed improvements in mitochondrial function, reduced oxidative stress markers, and enhanced muscle performance. This represents one of the first demonstrations of mitophagy activation in humans, a direct intervention in cellular cleanup. But there is a complication. Not everyone produces urolithin A efficiently. The conversion depends on specific gut bacteria, and only about 30 to 40% of individuals have the microbial composition required for strong production. For the remaining majority, pomegranate consumption may result in minimal urolithin A formation, which explains why results vary between individuals. This introduces a new layer to aging biology, not just what you consume, but how your body processes it. From a growing perspective, pomegranate is resilient. It can be cultivated in a range of climates, produces heavily once established, and offers a seasonal supply of its precursor compounds. But its full effect depends on internal conditions, the microbiome, which turns this plant into something more than a nutrient source. It becomes a collaboration between plant chemistry and human biology, and when that collaboration works, it activates one of the most precise cellular repair systems known. And the next plant shifts focus to metabolic control. One of the most powerful drivers of aging is rarely discussed in these terms. It is not a compound, it is a signal. The insulin pathway. Fenugreek targets the system directly. Derived from Trigonella phonum graecum, this plant contains compounds that influence how the body processes glucose. And this matters at a fundamental level. The insulin IGF-1 signaling pathway is one of the most conserved longevity pathways in biology. Across species, from simple organisms to mammals, reduced activity in this pathway is consistently associated with longer lifespan. The reason is structural. When insulin levels remain elevated, the body stays in a constant growth state. Cells prioritize expansion over repair, and over time, this accelerates damage accumulation. Fenugreek interrupts this pattern. Its soluble fiber, galactomannan, slows the absorption of glucose after meals. This reduces blood sugar spikes and lowers the corresponding insulin response. A smaller spike, a shorter signal, less long-term activation. There is also a second mechanism. A compound called trigonellin activates AMPK. AMPK is often described as the cell's energy sensor. When activated, it shifts the cell from growth mode into maintenance mode. It promotes repair, enhances metabolic efficiency, and overlaps with the same pathways triggered by exercise and caloric restriction. This makes fenugreek a metabolic regulator, not through force, but through modulation. A third layer is anti-glycation. When glucose binds to proteins or DNA, it forms advanced glycation end products, AGEs. These compounds stiffen tissues, disrupt cellular function, and contribute to visible and internal aging. Fenugreek has been shown to reduce glycation in both experimental and clinical settings, which means it not only controls the signal, it limits the damage caused by that signal. From a growing perspective, fenugreek is immediate. It grows quickly, sprouts in days, and can be harvested as both microgreens and mature plants. This creates flexibility. Daily consumption, minimal effort, continuous supply, because the goal is not occasional intervention, it is consistent regulation. Keeping one of the body's most powerful aging signals under control, not eliminating it, but preventing it from dominating. And the next plant works at an even deeper level, repairing the blueprint itself. Aloe vera is far more than a houseplant. Inside its thick, succulent leaves is a treasure trove of molecules that influence fundamental aging mechanisms. Chief among them are Axeman and polysaccharides. These compounds directly stimulate DNA repair pathways, enhancing the cell's ability to correct damage before it becomes permanent. This is critical because DNA damage accumulation is one of the primary drivers of aging. When the blueprint itself deteriorates, everything else, mitochondrial function, cellular division, immune response, begins to falter. Aloe doesn't just repair, it also supports the building blocks of tissue. Collagen synthesis, the process that maintains skin elasticity and structural integrity, naturally declines with age. Internal consumption of aloe gel has been shown to increase dermal collagen production, improving skin elasticity in clinical studies. In other words, aloe targets aging at two levels: the genetic blueprint and the structural framework of tissues. The anti-inflammatory angle is just as important. Aloe's bioactive compounds modulate immune signaling, reducing low-grade chronic inflammation that accelerates cellular aging. This makes aloe a multifunctional anti-aging plant, addressing mechanisms rarely tackled by diet alone. Growing your own aloe is simple and effective. A mature plant in a pot provides a continuous supply of gel. It requires minimal watering, tolerates indoor conditions, and thrives in indirect sunlight. By harvesting fresh gel, you maximize bioactive compounds, often far more potent than commercial processed products. Unlike capsules or gels that sit on a shelf for months, freshly extracted aloe contains active enzymes and polysaccharides in their natural matrix. This is the difference between a plant as a nutrient source and a plant as a cellular intervention. Daily small doses can complement other anti-aging strategies, from NRF2 activators like broccoli sprouts to insulin-modulating fenugreek. As we have seen, the synergy of these natural compounds is truly remarkable. And next, we'll explore lavender, a plant that works on stress, sleep, and neuronal protection, the overlooked pillars of longevity. Lavender, the fragrant purple herb, isn't just calming to the senses. It addresses three underappreciated anti-aging mechanisms: cortisol reduction, neurological protection, and sleep quality enhancement. Chronic stress accelerates aging at the cellular level. Elevated cortisol shortens telomeres, promotes mitochondrial dysfunction, and triggers inflammatory pathways. Lavender's volatile compounds, such as linalool and linaloyl acetate, modulate these stress pathways. Clinical trials demonstrate measurable reductions in cortisol after exposure to lavender aroma, even in high-stress populations. But lavender goes further than stress relief. Its neuroprotective effects support brain health by reducing oxidative stress in neurons and enhancing neural repair processes. This is particularly relevant for the hippocampus and prefrontal cortex, regions most vulnerable to age-related decline. Finally, lavender promotes restorative sleep, a critical, yet often overlooked, anti-aging process. Deep sleep is when the brain clears metabolic waste, DNA repair peaks, and growth hormone stimulates tissue regeneration. Studies show that exposure to lavender aroma before sleep improves sleep efficiency, increases slow-wave sleep, and reduces nighttime awakenings. In short, lavender enhances the cellular repair systems that run while you sleep. Growing lavender is straightforward. It thrives in zones 5-9 as a perennial and can be grown in containers in any zone. Incorporating lavender into daily life is a practical, evidence-backed intervention. Next, we'll cover Japanese knotweed. Japanese knotweed, Reinutria japonica, contains some of the highest naturally occurring concentrations of resveratrol on Earth. Resveratrol is a polyphenol famous for activating the SIRT1 longevity pathway, mimicking some effects of caloric restriction, promoting DNA repair, mitochondrial function, and anti-inflammatory pathways. While its molecular effects are well documented, Japanese knotweed is one of the most invasive plants in the United States, capable of forming dense monocultures and damaging infrastructure. Because of this, growing it yourself is generally discouraged. Instead, existing naturalized populations can be harvested responsibly, or commercial supplements labeled as Japanese knotweed extract provide the active compound without ecological risk. The SIRT1 mechanism. SIRT1 is a protein that regulates multiple cellular longevity processes, including mitochondrial biogenesis, DNA repair, and suppression of pro-inflammatory signaling. Activation of SIRT1 is strongly correlated with delayed cellular aging in model organisms. Resveratrol from Japanese knotweed triggers this pathway, producing measurable benefits at the cellular level. Bioavailability considerations. Resveratrol is rapidly metabolized in the gut and liver. Human trials show mixed outcomes, partly because only a fraction of the ingested compound reaches systemic circulation. Supplemental forms or formulations designed for improved bioavailability may deliver more consistent results than dietary consumption alone. Ethical harvesting approach. If knotweed is present locally, harvesting young shoots in spring is one of the few interventions that can both provide benefit and help reduce plant vigor. Young shoots are edible, with a tart flavor similar to rhubarb, and contain the highest concentrations of resveratrol. Otherwise, purchase ethically sourced extracts from reliable suppliers. While Japanese knotweed is not a plant most gardeners will cultivate, it is a highly potent longevity compound. Next, we explore moringa and its anti-aging benefits. Moringa, Moringa oleifera is often called the most nutritionally complete plant on Earth. Its leaves, seeds, and pods are rich in vitamins, minerals, protein, and polyphenols, making it a powerful anti-aging food at both the cellular and systemic level. When compared gram for gram, it outperforms many common health foods in essential nutrients like vitamin A, calcium, and iron. The primary mechanisms behind its power include potent antioxidant and anti-inflammatory effects. Specific compounds like flavonoids and chlorogenic acid actively reduce oxidative stress and chronic inflammation throughout the body. Isothiokenates like moringa trigger the NRF2 pathway, activating cellular defense genes that enhance DNA repair and mitochondrial protection. Then there's the induction of a hormetic stress response, a mechanism often overlooked in the world of longevity. Low-level stress activates protective pathways that improve long-term cellular function, effectively strengthening the cell's resilience. Moringa's bioactive compounds act as dietary hormetins, stimulating AMPK, SIRT1, and NRF2, all central regulators of human longevity. It's not just for the body. Moringa offers powerful neuroprotection as well. Studies show it increases BDNF production, improving hippocampal function and neuroplasticity, much like the anthocyanins found in blueberries. If you want to grow your own, it's remarkably easy. In zones 9 through 12, you can plant it directly in the ground. For cooler climates, container growing works perfectly, allowing you to bring your tree indoors during the harsh winter months. It's a fast-growing tree that rewards you with multiple harvests per year. Just ensure light, well-drained soil and regular watering. Moringa is a living, sustainable source of potent anti-aging compounds, providing multiple cellular benefits in one incredible plant. Next, we will cover ashitaba, the Japanese longevity herb that activates autophagy. See you then. Ashitaba, known as tomorrow's leaf, is a Japanese herb famous for its rapid regeneration and powerful anti-aging properties. Its secret lies in a compound called DMC, which activates autophagy, the body's natural process for cleaning out damaged cells. This cellular housekeeping removes dysfunctional proteins and mitochondria, promoting long-term health and tissue resilience. Studies suggest that by restoring autophagy, ashitaba could be a significant plant-based tool for extending our health span. The leaves are also packed with flavonoids and antioxidants, which support both your heart and metabolic health. You can grow it yourself in zones 7 through 10 or in containers indoors. It loves full sun and well-drained soil. When harvesting, look for the yellow sap. That's the sign of active compounds ready to boost your longevity. Ashitaba is truly nature's gift for rejuvenation. Start your journey with tomorrow's leaf today. These 15 plants are the ultimate botanical tools for cellular longevity. Superfoods like green tea, turmeric, and moringa support telomeres and mitochondrial health, while blueberries and lavender protect neurons and reduce stress. Growing your own ensures peak freshness and bioavailability, often surpassing commercial supplements. Incorporate them daily. Steep leaf teas, blend fresh fruit into smoothies, or harvest nutrient-dense sprouts. This garden-based approach targets everything from autophagy to inflammation control. Whether you have a patio or a full garden, you can take control of your cellular health today. Subscribe to Gardening Wizard, ring the bell, and tell us, which plant will you grow first? Your choice could transform your health at the cellular level.